Well, thanks everyone, and welcome to the session number 6, the Microbiology Aware Design of Space Vehicles, Part 2. So I'd like to introduce Dr. Rachel Adams from the University of California, Berkeley, where she'll be giving a talk on persistence, predisposition, and potential evolution of space. Rachel?
So I should say this is potential evolution in space, not that's another level about evolving space. Thanks to the organizers for inviting me. I'm happy to talk to you guys about I guess I should say what was asked of me to talk. So the invitation was talk about evolution in indoor environments. And my first thought, well, that's very interesting. Problem is, that's not what I do. But it's you know, and I don't know many other people that do. So it's a relatively sort of unexplored area of research. And so what I'm going to do instead is talk about, from sort of an ecological perspective, what are the processes that we would expect around something evolving in space and sort of the conditions around that. And so those ecological processes that I'm thinking about are predisposition, so the idea of certain microbes being pre-adapted or predisposed to exist in certain conditions. Second is persistence, so the ability of microbes to remain viable in trying circumstances, and then trying to understand and improve methods for which we evaluate what are the conditions that would allow them to then proliferate or potentially even evolve into another life form that we may particularly care about. So first is predisposition. And this is, the idea is that these organisms have evolved to live somewhere. And sometimes the leap from their natural habitat to indoor environments is not really a leap at all. It's not very creative. So the same material, cellulose that's in plants, we bring that material into our homes in the form of drywall, where we have paper that sandwiches gypsum board. And the best story I've heard about this is why this is a problem is not even the dumbest of the 3 little pigs built his house out of paper, which is what we do, because as we learned from Karen, when these things get wet, it's very easy for microbes to grow, fungi to grow. And then another sort of direct connection is surfactants. So surfactants are oils on leaf surfaces, and when there's runoff in streams, you get these blooms that look like someone dumped detergent in the stream, but those are natural chemicals, and then those same chemicals we bring into our homes in the forms of soaps and shampoos, and unsurprisingly, those fungi end up in our showers. But there's other situations that require a little more creativity in thinking about how did the organism sort of evolve a predisposition to be there. And one of the few examples I know of is this organism called Exophiala dermatitidis, which people care about because it's an opportunistic pathogen. And this is an organism, a fungus, that can actually survive inside a dishwasher. So it survives the conditions of dishwashing. And so people asked, where did this come from in the environment that it's now able to grow inside our dishwashers? And so, of course, they had some ideas, but they went to Thailand, and at first they sampled human-associated habitats. And they found it quite common in places that are associated with human habitats that get really hot, so sauna facilities and railways. And when they were looking in natural environments, they found it, but sparingly, in places like on the skins of wild fruits and berries, in animal feces, a little bit in water, and then a little bit in soil. And given the conditions that this organism could grow, so it can have different growth forms. It can grow as a yeast. It can grow as a filamentous fungus with hyphae. It can grow meristomatically. It's a leucotropic, so it's really sort of crafty and surviving in low nutrient conditions. It can handle extreme environmental stress like high temperature, low pH, and it has strong adhesion properties. And so what they hypothesized is that this is a yeast that's associated with frugivorous animals. So it's a yeast that grows on the skins of of fruits. A mammal, like a bat, will eat the skin. The fungus will survive the passing through the gastrointestinal tract of the fox and to be deposited in the feces onto the soil, and then the cycle conditions again. So the idea is that this is passing through digestional tract is like passing through a dishwasher. And so we found organisms that are sort of pre-adapted to our built environment. And Exophiala is an example of a black yeast that I think is one that we should be thinking about in terms of evolution in space environments. So black yeasts are a very broad term. They're found in soils and leaf surfaces, but they're also found in more interesting environments like associated with ants. They're actually a pathogen on mammals and then can infect brains. And then they actually will grow on mineral surfaces. So you can find black yeast growing on glass, just growing on rocks. So this is a really sort of a hardy type of organism. And they're found across diverse phylogenetic lineages. So this is a sort of a non-exhaustive survey of where black yeast are found on the fungal tree of life. Here we're looking at orders, and then within 3 classes, they're particularly abundant and common. But in general, it's sort of a broad taxonomic group, black yeast. And so if you're just kind of trying to get a back of the envelope idea of what organisms do we think would be likely to evolve, to survive, proliferate in an indoor environment and evolve, you might develop a list. And this list might be things like it's UV tolerant, it's oligotrophic, stress tolerant, it's probably melanized, really hardy spores that can persist, and drought tolerant. But then the problem is you get data and all this goes away. So this is an example of some work where this is, I mean, other people in the room can tell you more, but this is a study where they actually put spores of microbes on the outside of the International Space Station for several years. And so after going on the tour, it seems like we saw the Japanese wing where this would have happened. And what they found is that on the outside, these spores can survive outside the ISS for 2 years with only a moderate decrease in survivability. And the ones that were shadowed, they couldn't find any sort of decrease in survivability. And this was particularly interesting because this is a fungus that we would think of as being pretty weak. So you can see it's not melanized, it's green. The spores are sort of single-celled spores, not particularly hardy at all. So the idea is that if we're thinking about the organisms that are going to make it to space, and I think we all have this idea, as said in this paper, that we're selecting for organisms that are most fit to survive long journeys in space by those are the ones in clean rooms and those are the ones that make it. The potential pool is actually quite large and diverse of what that is, those kind of organisms. And on some level, it may not actually matter what kind of organism it is. So Charles was talking yesterday about how knowing the identity of the particular cause of that pneumonia was important for treating it. But in the indoor environment, that's not true. So if this was your bedroom and you went to an industrial hygienist and said, what do I do? They don't say, well, let me tell you what it is first. Oh, it's not Stachybotrys, it's Cladosporium, carry on. Like, that doesn't happen. They just tell you to fix that, right? We don't actually need to know what it is. And so the understanding the function, in this case, health effects, is more important than understanding the identity. And there's an interesting work going on at the Air Force Research Lab where they're specifically looking at the function of polyurethane coatings, polymeric coatings on metal surfaces. And they have, so it's a big issue where they have microbial growth on their aircraft. And so they can do surveys, they find a large taxonomic and phylogenetic diversity of the microbes. And then when they actually look at which ones are capable of a specific function, they find, so of these that actually grow, every single group had at least some that were capable of degrading these compounds. So if we sort of accept the fact that the idea that there's organisms pre-adapted to do really well in space, and that's broad, the questions sort of become, in order to care about them, are they alive and are they doing anything? And so NASA has done a lot of work in trying to get this connection between sequence-based detections and are they alive. So I won't go into that, but I'll briefly mention some work that we've done looking at indoor environments, where we all know that when we sequence indoor environments, we find a lot of human-associated bacteria. And my hypothesis before this was that this is sort of a signal of microbial or skin detritus. So there's lots of skin flakes everywhere. Most of these things are alive, are dead. And we're sort of seeing a signal of past desquamation. And so someone in the lab did a simple project where they're asking how long after skin shells are shed do the bacteria and fungi on those shed skin shells remain viable. Does anyone want to have like a guess, sort of a benchmark about how long you would think? Huh? Weeks. On the what? So, okay. So what we did to ask that was we collected skin shells in the form of tape lifts, and then let them sit in sterile conditions where they're not getting inoculated. And then at every 2-week intervals up to 10 weeks, simply plate them and ask what would grow. And so we did this. Oh, and this is just to show that this is sort of normal built environment conditions. It's not particularly moist, not particularly warm. So this is kind of the conditions that you would expect in most built environments. And then this is an example of what a plate might look like. And we did it on 3 different medias, and we plated every 2 weeks. And so we had 10, in the study, we had 10 volunteers. And so what we're looking at is the 3 media types, and then the percent coverage. So basically how much of the media plates are covered with the microbes. And so you can see there's variation. But overall, you see what you expect to be a declining trend over time. But interestingly, at 10 weeks, we hadn't sort of reached the limit of where we couldn't get any of these microbes to grow. So at 10 weeks, only 2 of the 10 individuals had no microbial growth on either of the 3. Right, and so we are doing the genetic analysis of the identities. But this was just a simple question of, can these things survive and sort of how long? When do you expect to see sort of the microbes on these skins to not be viable anymore? So this was surprising to me. Well, first of all, it was like surprising that you couldn't find data on sort of how long these things would remain viable. And then the time frame was surprising to me. So understanding sort of once they're there and are they alive, are they actually doing anything? And in what conditions are they doing it? And that's sort of a big open question in all indoor environments. And so we all know that we can design buildings perfectly and execute their building perfectly, but buildings fail for the very simple reason that water ends up where we don't want it to be. And so the question, a lot of times from studying microbiology of the built environment, is not necessarily what grew, but what are the conditions that would lead that, and how can we know early so we could potentially stop it. So sort of early detection of building failure is a really important component of protecting public health. And there are three avenues or ways that I've seen people trying to get at this. And so one is, and I'll just mention all of these are based not on discrete sort of monitoring events, but are continuous in some way. So we're sort of avoiding the question of how often do we need to monitor, because all these are based, they're integrated, I'll show you. So first are meters that we've developed to just as single time events to tell you how much moisture is in a building material. And Karen was mentioning this idea of water activity, and this is the metric that really matters in a building material because it's telling you how much water is available for the microbes. Now the issue with this kind of device is that when you actually, it's a device that you sort of put on a surface. And then the problem is when you do this, it takes a really long time to give you the information you need. So we did this where we put a sensor on a piece of gypsum board that had a high water activity. It took a day, a full day, to get to that level. So that's really not practical for monitoring buildings. So there's a lot of potential innovation that can happen because the idea is there. If we know the water activity material, we can tell you if microbes are going to grow to a good first approximation. So what needs to happen is the implementation of this in buildings in a way that can be tracked. So there's been another sensor that's sort of biologically derived, and this is called an Abe sensor. And so what they did is they put spores in a nutrient source on a film, and then you put this film on the building material and leave it in your building. And then you come back in 6 months and you ask, did anything grow? And the advantage of this is it's actually getting the exact variable you want. Is microbes are going to grow on that surface from the water, and it's catching intermittent growth. So sometimes water sources are not constant, right? They're dependent on the situation. And so this kind of sensor will pick up that intermittent water availability. And there's been some work sort of following up on this where they use that sort of surface, but then they put a device over it called the Mould Spy, and it's sort of measuring how much mold is growing and sort of giving you a signal in some sort of central place. And so you can sort of monitor, the idea is you're monitoring different places in your building. The last one, or the third way, is potentially chemical sensors. And so this is work that Karen mentioned. It's the we call it CHEM, the Environmental Microbes and Moisture Study, where we're asking what's microbial activity indoors under sort of realistic building conditions, and what's the sort of chemical signal associated with that? What chemicals are the microbes emitting under these different conditions, and what kind of chemistry are they doing? And so this work is currently ongoing. So I'll just give you some preliminary observations. One is that people very much trust their vacuum cleaner. So when we ask people to send us their vacuum dust, we get really kind of rich communities to play with. Another thing is one of the big challenges in this project is sort of understanding the microbial signal from the purely chemical signal. And so one of the things that we wanted to do was have sterilized dust to play with. And we knew the autoclaving wasn't going to be good enough to get a sort of a sterile dust signal, so we went to gamma radiation. And so we radiated dust from a local facility, and the bottom is what, if you just throw your house dust on a petri dish, you get a nice flora. And you see, even in the radiated dust, it's not completely sterile. So it's definitely diminished, and it's definitely reduced. But you really have to play around with your sterilization technique to get a purely sterile house dust sample. It was only 10 kilogray. Okay, and then in preliminary work, we are seeing some volatiles that are likely microbially derived are changing with the RH conditions. So this is an instrument that can detect a lot of volatile organic compounds in the concentration of parts per trillion in real time. And so we can do things like incubate these samples and change how much relative humidity in the chamber that they're in and sort of track to see what the chemicals that are coming out of. And you see some of these, like acetaldehyde and butyric acid, are responding to changing RH. So they're increasing during high humidity and decreasing and diminishing during low RH. And then you don't see a signal with compounds that are presumably not microbially derived. So this is over time, and what it is, it's different relative humidity of the air that's going through the chambers. It just changes instantly. Like one sniffing period is 85 and the next it's 50. So then the last question of if they're there and they're alive and they're persisting, will they evolve? And I think this is a really interesting question, and I'm thinking about this in the context of building-associated microbes evolving to something that we care about. And evolution to sort of human-dominated habitats is not well documented, but it's sort of a growing field. And there's some interesting examples. So animals evolving tolerance to pollution, or hawksbeard is an interesting example. It's actually evolved its dispersal to be less dispersal ability, because the idea of it's growing in parking or sidewalk squares, you don't want the spores the seeds going very far. You want it to fall right where it is. The white-footed mouse has basically evolved in New York. There's evidence that's evolving to eat moldy peanuts, because that's sort of what people drop. So there's some interesting work, but if you notice that none of these are microbes. So right now we just sort of have an idea of how would microbes evolve to indoor environments. And I think that a barrier to understanding that in indoor environments is that most microbial colonization on buildings are random and repeated colonizations from nature. So if you have fungus growing in your home and your neighbor also gets a water leak, it's way more likely that whatever colonizes your neighbor's home blew in from the window rather than coming from your house. So the opportunities from sort of long-term evolution of a moldy home are not very high, I would say. But in terms of how they would evolve, you can think they might involve interesting dispersal mechanisms. So mushrooms do have gravitropism, where they orient themselves, and so you can imagine there's going to be some interesting processes there when you grow them in zero gravity, UV radiation. But I think an interesting one is the evolution to infectious agents or to pathogens. And there's been sort of a model for this. And so what they were positing is that you might have an organism like Exophiala dermatitidis or Bacillus pullulen, so like yeasts that live in homes, that these are polyextremotolerant. So they've evolved to have resistance to many stress vectors. And they may infect an immunocompromised host, but that's sort of a small part of their ecological repertoire. But then you get this evolution from this polyextremotolerant state to something where it's, well, first of all, you can go one way where it sort of becomes specialized on a particular stress factor. Or you can go the other way where it first becomes more of an opportunistic pathogen, where it's spending more of its time infecting a host. And then even more so, that it's even sort of able to infect an immunocompetent person to get an obligate pathogen. So this is sort of a model for how you go from an organism that's evolved to survive outdoors in extreme conditions to being an organism that's sort of an issue for humans. And there's a couple reasons to think that, again, these black yeast lice fungi are particularly ones to care about. So they're melanized, they have pleomorphic growth, they can sort of switch their life form based on the conditions that is happening. They do grow in biofilms, and they're strong emitters of exopolysaccharides. So at this point, sort of the idea of evolving into indoor environments is more of a model than data-driven. But lastly, I'll introduce the idea of microbes in space, but they're on purpose as a food source. So when we think of microbes, mushrooms, you might think of a nice mushroom. But this is the fruiting body, and a sort of a very inefficient way of eating mushrooms. Sort of a much more efficient way is actually eating the hyphae. And this is exactly what's done if anyone's eaten corn. What it is, it's a Fusarium species that they grow, and it makes sense on theory. So you take compost. This could include human excrement, paper, any sort of wrappings you have, and this organism will turn that compost into hyphae, which you can then eat as a nutrition source. So just to sort of link the two topics of microbes in buildings and food production in space, throwing this out there. And last, I'll acknowledge my collaborators, and thank you for listening.